Mind the Plant is a one-of-a-kind laboratory where researchers with diverse expertise such as bioinformatics, bioengineering, analytical chemistry, philosophy, plant physiology, as well as physiological and comparative psychology, study plant behavior. This laboratory is funded by the European Research Council through the Rumors Project, awarded to Professor Umberto Castiello. We diverge from the position that plant knowledge is useful solely for optimizing crops. The idea is to study plants as individual organisms, equipped with a complex sensory apparatus that provides information to respond and adapt to a continuously changing environment. We aim to understand how plants live their lives by investigating how they contextually modify their individual and social interactions during development. One way to approach this multi-level research is to jointly analyze the kinematics of their movements, the type and quantity of emitted organic molecules, the study of electrical potentials, and gene expression. Mind the Plant is a custom-made, fully automated laboratory, a tailor-made suite designed carefully from the simplest elements to the most sophisticated instrumentation. It consists of a climate chamber served by two air handling units, both equipped with a pre-filter, air filter, fan, and sealing recirculations. Inside the climate chamber, there are 8 grow chambers, each of which is monitored and operated remotely from a control panel. Each grow chamber allows control over temperature, humidity, ventilation, lighting, and irrigation systems. Data acquisition and monitoring of what happens in each growth chamber occur in real time without interfering with ongoing data acquisition. For the three-dimensional analysis of plant movement, each chamber is equipped with 2 infrared cameras that allow uninterrupted day and night observation of their movements. Electrodes are placed on the plants to monitor changes in electrical activity. To study the biochemical behavior of plants, we focus simultaneously on volatile organic compounds, commonly known as VOCs, emitted by the plants. These are small carbon-based chemical molecules that evaporate extremely easily. They are found in a wide range of natural substances and play an important role in many aspects of plant life, especially in communication processes. To study them, we use a time-of-flight mass spectrometer, which allows continuous and real-time monitoring of volatile organic compounds, even at low concentrations. The volatile molecules are ionized and transported via a nitrogen flow to a detector, where the mass of the charged particles is measured to determine their identity. The nitrogen flow is provided by a generator composed of 2 modules. The first module captures and compresses atmospheric air, which is about 80% nitrogen, while the second module filters the compressed air to obtain 100% nitrogen. Finally, we will use a second mass spectrometer to more precisely identify the detected molecules. Roots are also closely monitored. Their movements are studied with microcameras placed inside the supports available for plants to cling to and grow in height. Complementing this monitoring is a system that allows for the study of VOCs emitted by the roots. The different experimental configurations allow repeated study of plants both in individual and social conditions. That is, more than one plant in the same pot or in different pots. The simultaneous acquisition of all this information generates a huge amount of data, which we manage to channel into a state-of-the-art server with high computing power. This allows us to store and reprocess every piece of data in real time without affecting or interrupting the monitoring. The server hosts software that enables the processing of collected data, its engineering and contextual analysis. Research continues outside the growth chambers, in the control room and the technical room, where equipment is located for preparing material and samples for morphological and transcriptomic analysis. Correlating the data collected through this parallel analysis will help to shed light on the evolution of cognitive processes in non-neural organisms, particularly on the different communication and interaction strategies of plants, with possible implications for understanding their ability to adapt to climate change.